Okay, I wanted to get together with you and do a video for you to kind of review some of the activities that you've done so far in the MAPS program. So hopefully you've spent some time with the water cups and then also used the phospholipid and membrane kit to understand how those water properties and how the properties of phospholipids act together to help form cell membranes. And so we are going to now review all of that, also using some molymod phospholipids. All right, so what I want to first start with is talking about the basic structure of a phospholipid molecule. There are many different kinds of phospholipids, and they have different functions in the cell, actually. But there's certain characteristics that they have in common. So let's talk about some of those things. So when we look at this phosphatidylcholine, we see that there is a glycerol backbone. So that is something that's standard in most phospholipids, is this 3-carbon glycerol backbone. So the 3 carbons are connected to 2 fatty acids and 1 head group. So what fatty acids are just long hydrocarbon chains. So long chains of carbons and hydrogens. And they have at their end a carboxylic acid group. And so a carboxylic acid is just this carbon that is bound to a carbonyl group, which is double bonded oxygen, and a single bonded OH group. So this is the carboxylic acid, and this is the carboxylic acid, and then this is the hydrocarbon chain of the 2 fatty acids that are connected to the glycerol. Now you'll look at these fatty acids, and you'll notice that they look different. Okay, this one is very, very straight. That is because it's what we call a saturated fatty acid or a saturated hydrocarbon. That means that every carbon in it is connected by single bonds. Okay? On this side, we see an unsaturated hydrocarbon or unsaturated fatty acid. And that is because it has a double bond. In this case, one of the bonds happens to be a double bond. There can be more than one. So it can be monounsaturated or it can be polyunsaturated. When a double bond exists in a hydrocarbon chain like this, the next carbon can bind in one of 2 places, okay, on this carbon. If the next carbon comes in and binds in this position, it's on the same side of this double bond, and that is called the cis configuration. And you'll notice that when it does that, that's when you get this kink. And so this is a cis bond. It could also bind in the other position. So that is on the opposite side of this double bond. And that is the trans position. Now most fatty acids that naturally occur in the body are in this cis configuration. So a number of years ago, back when I was a kid, the food industry decided that they thought it would be a great idea to try to make some chemical modifications to the oils that are in our food to try to make them solid at room temperature. And so they hydrogenated oils in order to make margarine and other solid fats. And what they did was they hydrogenated and made them into this trans configuration, and that allowed them to be solid. And the problem with that is we've found out now over the years that this can lead to a lot of health problems. It increases the bad cholesterol, the LDL cholesterol in our bodies. It decreases the good cholesterol. And if it gets inserted into our phospholipids, into our cell membranes, it's going to make more of our fatty acids like the saturated one, linear like that, and less kinked. Okay, why is that a problem? Well, we'll talk about this a little bit more later, but the reason that this kink is important in our cell membranes is to maintain that fluidity that we said is really important. So these fatty acid tails, we said, can be saturated, are usually one saturated, one unsaturated. Okay, so depending on where the cell is, what tissue the cell is in, what that cell's function is, these fatty acids can be different. They can be different lengths. They can have their double bonds in different places. They can have one or more double bonds. Very, very cell and tissue dependent. What makes a phospholipid a specific phospholipid, though, is not those fatty acid tails. It is the head group. So in this case, we are looking at phosphatidylserine, which is a very common phospholipid in the cell membrane. And all head groups in phospholipids have this phosphate group, all right? This phosphate group, it's a phosphorus that's bound to 3 oxygens, and then it's got the carbonyl oxygen as well, so a double bonded oxygen. And then that group is attached to the specific head group. And this happens to be a choline group. So this is phosphatidylcholine. Now, something that you will notice when we start looking at all the different phospholipids is that there are some charged atoms in this, okay? The phosphate group always has this oxygen bound here. And this oxygen is unique in that most oxygens have 2 bonds, right? 2 electrons that they bond to other atoms with. This one happens to only be bound to the phosphorus. So that allows it to have a negative charge. Now this nitrogen group in the head group has actually 5 electrons in its outer shell. And so it is bound to 4 carbons. It has it has a positive charge. So there's a negative charge here and a positive charge here. They cancel each other out. So it's still a neutral head group, but it is polar. It has these charges, and that polar nature makes it want to hang out with water. So these fatty acids, because they're all hydrocarbons, are nonpolar. They do not like to hang out with water. They are hydrophobic. And this is polar. The head is polar. It is hydrophilic. The fact that it has these 2 very different parts of this molecule makes it, we call that amphipathic, amphiphilic. You'll hear different terms, but what that means is that it's got both. It's nonpolar and polar at the same time. So now we're going to take this information and do the water beaker activity that hopefully you guys have already done, and see how these properties help form the structure of cell membranes. Okay, so let's review the water beaker activity that you all have gone through already. So we have a beaker of water, and we have these little representations of the phospholipids that we just talked about. You see the polar heads are in red, and then the fatty acid tails are in yellow. So what would happen if we added some of these phospholipid molecules to the surface of the water? Hopefully you'll realize that because the hydrophilic heads, or the polar heads, like the water, they will want to be down. And these hydrophobic tails, the fatty acids, will not want to be in the water. And so they will arrange themselves with their tails sticking up out of the water, like so. Now let's assume that we want to actually submerge these molecules in the water. How is that going to happen? So let's do that. Now we know that the hydrophilic heads are going to be fine in the water, but the hydrophobic tails are not. So what they're going to do is trying to avoid the water, and they find other like-minded portions of the molecules. So all the hydrophobic tails will tend to want to be near each other, and the hydrophilic heads will want to be near the water. So that is how they would orient themselves. Now that's called a micelle, basically a lipid droplet. Okay? Now, let's try to assume that we want to have a micelle that actually has water on the inside. How would we make that? So let's make it a little bit larger so that we can put a few water molecules in there so that we know that that's what they're doing. And now how will we make that work? Now we've got those tails that are next to the water, and they're not going to like that. So what we need to do then is let those hydrophobic tails be near each other again. And in order to do that, we will allow them to form a bilayer, because that way the phosphate heads, the polar head groups, can still be near the water on both sides, inside and out, while the lipids are facing each other. So this is a representation of what the cell actually looks like, what the cell membrane looks like, because all cells are submerged in water. They have water on the outside in the extracellular space, and they have water on the inside in the cytoplasm. And so by allowing phospholipids to be a bilayer and have the polar head groups on the outside as well as the inside, that allows it to be in that type of environment. Okay, let's go back to the molymods and review what we talked about with the phospholipid structure, and then start to look at some of the differences between some of the phospholipids and what those mean. Now, again, we said that the general form structure of these things are that you have this 3-carbon glycerol backbone, and then you've got 2 fatty acid chains. One's usually saturated, one's usually unsaturated, and then there's a phosphate head group. Now, this is, remember, phosphatidylserine. We talked about that before. The head is what determines what phospholipid it is. The fatty acids are variable, so they can have different lengths. They're usually between about 14 carbons and 24 carbons. I think I'm around 17 or 18 I made these. So this is kind of medium or even on the small side. So they can have very, very long fatty acids. The fatty acid content is dependent upon where it exists in the body, what cell types, what the function of the cell is. So that the different fatty acid lengths really have a lot to do with its particular function. Now, this is one other very common phospholipid in the cell membrane. This is phosphatidylcholine. This is called phosphatidylserine. So the difference here, everything else is very similar. Like I said, the fatty acids can vary. Where the double bond is can vary, but that's not important. What's important is the head group. In this case, we've got a serine head group. And the serine head group is kind of special. And we're going to talk a little bit later about some of the special functions that phosphatidylserine has because of its special structure. Now, we talked before, in review, about this oxygen that had a negative charge and this nitrogen that has a positive charge, and how that they balanced each other out so they were overall a neutral head group. Well, phosphatidylserine is different. So it has that same negative charge with this oxygen. It's got that same positive charge with this nitrogen group. But it also has this carboxylic acid group. Remember, we talked about the carboxylic acid group, which is the carbonyl double bonded oxygen and the single oxygen here. This oxygen, as you can see, again has one bond, and so it is also negative. So there's a negative charge here and here, and a positive charge here. That gives phosphatidylserine an overall negative charge. All right? Now remember that. We'll come back to that a little bit later. But again, it's a polar head group. So that's the most important part as far as cell structure goes. Now, if you decarboxylate this, phospholipid, which basically means you just take off that carboxylic acid group. Then you get a different phospholipid, phosphatidylethanolamine. Okay? So phosphatidylethanolamine, phosphatidylserine, phosphatidylcholine. That makes up 3 of the most common phospholipids. So here we have phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, and now we are going to talk about the 4th type of phospholipid, sphingomyelin. Now, sphingomyelin is a little different. So, unlike the other 3, sphingomyelin has a different kind of backbone. In sphingomyelin, the backbone is not a 3-carbon glycerol, it is a sphingosine molecule. And a sphingosine molecule has 3 carbons, but this first carbon is bound to a hydrocarbon, and it also has, on the second carbon, it has a nitrogen atom here. So that's a little different conformation than the glycerol backbone. And then what the sphingomyelin molecule then adds is 1 fatty acid. So here we have that carbonyl group, and we've got the fatty acid tail here. Now, the structure of sphingomyelin is very cylindrical. So these chains are almost always saturated, very highly saturated and very cylindrical. This is common in all cell membranes, but it exists in larger concentrations in certain cells. So in the cells of the nervous system, the Schwann cells and the oligodendrocytes that make up the myelin sheaths, they are very highly enriched for sphingomyelin, hence the name. So they tightly pack. Their lipids, because they're very linear, they tightly pack, and they're specialized for certain types of cells, like those that form the myelin sheaths. Now, if we look at the head groups, though, you might see some similarities with what we have over here. That's a choline head group. So actually, the head group can be, and often is, the choline head group in sphingomyelin. So the head groups can be the same, but the backbone is different, and this chain is different. All right, so that's just another way of showing how these phospholipids vary slightly because they have different functions. Now I want to tell you one more story about that with using phosphatidylserine. Now phosphatidylserine, you'll recall, is special because it doesn't have a positive and a negative charge. It has 2 negative charges and a positive charge, which makes it net negative. Now this is important because it acts as a kind of a marker in the cell for certain enzymes. Phosphatidylserine is always located on what they call the inner leaflet of the phospholipid bilayer. So it is always, if I'm the cytoplasm, it is always facing in. And there will be other phospholipids that will be on the outer leaflet facing out. Now, how does that happen? Well, there are specific enzymes called flipases. And flipases will identify phosphatidylserine that is somehow has gotten on the outside of the cell, and it flips it back to the inside. It needs to be on the inside at all times. Now these phospholipids are more than happy. They can move back and forth and from one end of a cell to the other end of the cell very, very quickly on one leaflet. But they do not flip from one side to the other very easily because these polar head groups would have to go through the lipid part of the bilayer and get to the outside. So the flipases make sure that the phosphatidylserine is on the inside where it belongs. Now, billions of your cells die every single day by a process called programmed cell death or apoptosis. And that is because cells get old or they get damaged and they have to be targeted to be broken down and gotten rid of, okay? The way this happens is there's a very involved process, apoptosis, where there's a cascade of events where proteins called caspases are activated. And caspases do a lot of different things that are involved in the cell death process. But one of the things that they do is they inhibit this flipase from making the phosphatidylserine be flipped to the inside all the time. They also increase the activity of something called a scramblase. And what a scramblase does is it scrambles up the orientation of the phospholipids in the cell membrane, so they're facing in all different directions. And that means that many of the phosphatidylserines will be facing out at that point. And that's what they call in the literature an eat me signal, because that signals that this cell is dying and needs to be removed from the system. And so there are cell types called macrophages, which are like little mini Pac-Man, and they come around and they recognize this phosphatidylserine because of its negative charge, and they engulf that cell and break it down and remove it. So that's just one way where the specific structure of these phospholipids are responsible for very important functions in the cell. Now there's just one other concept that I want to talk about in regard to this phospholipid membrane. Now, we talked about the fact that these head groups are on the outside and the inside associated with water, and that the fatty acid chains are facing each other and form the inside of the lipid bilayer. And it's important that you have these kinks, because that allows this membrane to be fluid and be able to move, and so all the functions that happen can happen, because we need a lot of proteins to be embedded here that can move around. We need these to be able to move around. So these kinks are very important. However, if this is all we had, it would make for a very wimpy cell membrane, okay? It would be kind of like a soap bubble. It wouldn't have a lot of structure to it. So we need to have a way to make it more of a barrier, so that we can't have things just kind of floating in and out in between these floppy phospholipids. So we have something called cholesterol. This is cholesterol, and you can see it has this 4-ring structure. Okay, 1, 2, 3, 4. And that makes it very sturdy and not very flexible. And this has an oxygen here, and then it's got this non-polar section here. So this polar oxygen likes to insert itself with the polar head groups. And then the non-polar section inserts itself between these tails, and it gives it some structure. Now it's very short, so there's still a lot of tail here that maintains the flexibility, but it gives it enough structure so that it can keep ions and things out, it can keep water out, it can keep it can keep things from passing through easily, and it allows the cell to have its specific structure that is required of it. Now, because of all this, there's a need then for ways to get certain substances through this barrier. And we know that we need something for ions, and so there are things called ion channels, like this potassium channel, that allows for the specific passage of potassium through the channel. It sits in the membrane like this and allows ions to pass through. We also have what you guys are studying, right? The aquaporin, the water channel, and that also sits in the membrane and allows water to pass through, because otherwise it would never be able to get through all this non-polar fatty acids, right? So that's what this is for. It allows a pore for the water to get through. So hopefully this video allowed you to better understand phospholipid structure and how they work together in the cell membrane to form the phospholipid bilayer, and why it's so important for us to have specific proteins that allow passage of substances like ions and water through this barrier. Because, as you can imagine, with all these nonpolar fatty acids, it would be very difficult for water to pass through without some assistance from an aquaporin. All right, that's it for now. Back to your mapping.